another episode of Catalysts, a platform for the scientific community to connect, discover, and transform. This is your host, Kedar Aris, and today it's my pleasure to connect with Dr. Katherine Chiappinelli, an assistant professor at the George Washington University in Washington, D.C., in the Department of Microbiology, Immunology, and Tropical Medicine. Kate's primary area of research is cancer epigenetics and immunology. In particular, her lab is investigating the epigenetic changes in cancer and how the epigenetic drugs can reverse these, specifically focusing on the non coding regions of the genome and the tumor cell immune response. She has published in high profile journals including Cell and Nature, among others, and currently has over 27 publications with an H index of 17 and an I-10 index of 22. She is currently funded by the NCI and has been a past recipient of the prestigious K Award. With that, let's welcome Catherine to our podcast. Hello, Kate. How are you doing?
I'm well, thank you. Thank you so much for having me.
Absolutely. To begin with, can you briefly share with us your academic journey, starting from PhD to postdoc to becoming an assistant professor, and how you successfully managed those transitions?
Sure. So I applied to PhD programs my senior year of college. So I was actually double major in biology and music. And I had really gotten into undergraduate research, both being exposed to that at my college, but I also did a summer internship at NIH. So I was able to get research experience and get an idea that I really enjoyed that and wanted to pursue that. So I applied to PhD programs then, and I interviewed at several places, and WashU in St. Louis was my absolute favorite interview because I really enjoyed it was the faculty that were both in terms of their the research, but also they were quite invested in the students. And so I was really glad to join that program. Um, and then I was there for about 5 years working with Dr. Paul Goodfellow. And just, I mean, this may be a good message for everyone, but my training was really in cancer genetics. And I didn't do any immunology until my postdoc, and now I'm a professor in an immunology department, right? So a lot of our research interests can change over time.
Absolutely.
So I worked on non-coding RNA in endometrial cancer and uterine cancer. And then I wanted to do a postdoc in the area of cancer epigenetics. And again, reached out to several labs with the assistance of my PI at that time, who was really a great mentor to me. And I interviewed at a couple different places, and I really was excited about Steve Balin's lab. He is one of the kind of founders of the cancer epigenetics field, and I spoke to several of his former mentees, and I really appreciate his mentoring style. So I accepted the position there at Johns Hopkins, and there did more kind of cancer epigenetics, but also getting into cancer immunology because of the turn that my project took. And so was there for I think 4.5 years, and then there I actually I applied for an F32 award from postdoc fellowship. Definitely did not do well the first round, but was able to revise it and get it, which is also a good thing for people to hear, I think. And then right after, as we were kind of getting the big paper from my postdoc accepted, applied for the K99, and was lucky to get that on the first try. So then I did my faculty interviews that year that I had it, and then accepted this position and started here in January 2017.
Got it. Speaking of the K Award, did you plan ahead given the strict 4 year eligibility window, which also includes any resubmissions? What was that process like for you?
think, I mean, it's a really big challenge, I think, for postdocs, right? Because especially if you have a project that involves mouse work, the things can take a very long time to get out there. And you really need to have, I think you need to have a first author publication from your postdoc at least accepted before
Right.
you can successfully apply. So I was aware of it, but only because another postdoc in Steve's group wanted to apply for it. But they actually changed the eligibility window while I was a postdoc. Like they changed it, I think, from 5 to 4 or something. It was a year less, and they didn't grandfather anybody in. So I had heard him be very frustrated with this. And then I started to look up eligibility and say, what is my window? Because it's even the 4 year is even for the revised application. So that can take
Absolutely. And
a
that's
long
the
time to
kicker.
get in. Yeah. So it's hard. So I knew about it because of that. And then I definitely I knew people who had received those grants and then had a very positive experience in the academic job market and starting their independent lab. So I really wanted to compete for it.
That makes sense. Now, can you briefly share your experience navigating the faculty job search process? And how much of an advantage do you think was the K Award during that process?
Yeah, I think it made it easier. I'm assuming that I probably got interviews easier because of it. I don't really know. I wasn't on the other side of it,
Right.
but I actually didn't have the award in hand when I applied. I just put the score. I had the score. So I put it, which I recommend everyone does. If you have a competitive score, put it on your application materials because that people know the funding cutoffs for NIH, right? So you can always update them, right? So if I'm applying in like August and I'm being interviewed in January, then I had it by January, right? But I didn't have it in hand in August. So if you have an accepted paper, if you have a good score in NIH grant, put that in your application, or even if you've applied for it and don't have the score yet, it's really important. So I think it helped to get interviews. When I was there, I would say it didn't really change the initial. Often you have, it depends on the place, but when you give a talk on your research, what you have done, and maybe 10 minutes at the end of it on what you plan to do, and then often there's a whole separate chalk talk, as you said,
Right.
and that for a lot of mine was the first visit. Some places do that on the second visit. For a lot of mine, it was research talk at like the beginning of the day, chalk talk at the end of the day. Some places back to back, which is pretty brutal. But I think that's all the process of like they're trying to see how well you think on your feet. So for the chalk talk, I think it made it so much easier to have written the K99 ROO because I knew I had 3 aims in my ROO. And that, I mean, it's not everything you're going to do in the first 5 years or whatever, but you can really, you have a structure, you've already kind of workshopped this grant. And it just makes that part so much easier. So I feel like that was the most, even if I hadn't gotten the K99, I would have been so much more prepared for that part. And I felt like, to be honest, I was really choosing places based on how my chalk talk and interaction with the department and or for in this case the cancer center, the members were at the chalk talk, what questions they asked, how our interactions were, and just really that interaction.
Got it. And how long does that process typically take? When should someone who's thinking of making that transition begin the process of applying for a job?
Good question. Yes, I would say give it a year. I probably started submitting. Often the applications are rolling,
Right.
right? But often the job postings start to come out. I would say August, September. So you submit, but then I think most of my interviews were kind of February through March, and then offers in hand June, right, for starting in the next fall. So yes, I think a year is it's good to be conservative. And some places may have more accelerated timelines, but it does take a long time.
And when did you officially join GW?
I actually joined in January. So I had an offer to start in September, but I was expecting our first child in September. And so I actually stayed on my K99 in Steve's lab to finish up, take maternity leave, kind of finishing everything up in the lab. And then I began in January here. So that usually those are negotiable, your start dates. That's also something that's good to hear.
Well, that's good to know. Speaking of negotiation, what was the salary negotiation process like? Did you receive help or mentoring during the process?
I think that it is really difficult to be in a position where we have to negotiate, because any other training in our field, we have just, applied to grad school, gotten a postdoc. There's not much negotiating you can do about a postdoc, right? I felt like it was a really stressful situation to be in. And I purposely, so my PI is really, really supportive. My postdoc PI was super supportive. And so was my PhD PI, but both of them were so senior that they were on the job market 30, 35 years ago, right? So it was a very different experience, and they had limited advice they could give me. So what I did was I sought out peers that were, 5 years ahead of me, that had started their own labs, and I talked to them on the phone. I said, how much should I be negotiating? Can you give me an example of the Excel spreadsheet you use to ask for startup money, right? Like, because what they want to do is they want to say, like, what do you want your startup to look like? So that was really how and then I would speak to them about that and say, what should I be negotiating? Should I negotiate startup, salary, et cetera? Everything is negotiable. So that's the case whether or not you have a K award. My K award, in this case, my PO from NCI, he was incredibly supportive of me, and he wanted to see my offer letter. And he actually told me to put specific things in the offer letter. So your
great.
PO should be on your side in terms of this. So he actually really helped me kind of stand up for myself and really make sure that it was an appropriate offer, because they're really, what NIH doesn't want is for institutions to hire people with ROOs and give them less money than they would otherwise because of the ROO money. They don't want people to be penalized. So he was really careful about including language in that letter that then, because they'll give you the offer letter, you edit it and put things in it that you want, send it back, make sure they're okay with it. But he really helped me in terms of that, of saying, no, like, for example, if you have startup funding, if you want it for, it says 3 years here, he says you really should ask for it, but you can spend it with unlimited time.
And that's such a great point about the program officer being an amazing resource. And I can personally vouch for that as well. During my K99 application process, I sought out my PO to get feedback on my specific aims and whether the project was a good fit for the study section, and even getting feedback on the reviewer comments from the first submission.
Yes, and usually you have to initiate that, right? So I do think that it's your job as the person either applying for the grant, or if once you have the grant, to initiate a conversation with your PO. For example, I had asked mine to speak on the phone, and he actually said, are you going to be at the AACR meeting next month? Why don't we just meet in person? And that was really nice to actually just meet in person and kind of go over everything. I'll be honest, I did not speak with them about the application. I had a very short time to write my K award. So I just wrote it and submitted it. If I had revisions, I definitely would have. I've done that with I have a recent R1 that was scored well on the second submission. So I definitely, it's incredibly helpful. Yeah, thanks. It was good. So that has been, I've definitely reached out. And then actually, you're right. So for another application, I had reached out with Ames. This is a second R1. I had reached out with an Ames page to say, is this appropriate for the study section, right? So that's always a good idea. And I find that they often want to talk on the phone rather than email, to go over everything. So I'll often email them and say, please let me know if email or the phone is more convenient. But yeah, I've had very positive experiences, and everyone has been really invested in the research. They have a scientific background, they know the research. Exactly. And it's usually in the field of what you're applying to. I really felt that, especially my K99 one, really advocated for me. So it's very positive.
Well, that's good to hear. Speaking of grant writing, given your successful track record in applying for and getting grants, including the F32, K99, R21, and now the R01, has the process of writing grants become easier, or is it still just as challenging as ever?
Yeah, we'll see what the next one looks like, but I think that mostly it's been very positive. So NIH likes to see that you got a training grant, and then you got a K, and then you got an R21, right? So that's any response I've gotten from reviewers has been very positive. So that yeah, I only good feedback on that. But I think that the most important thing for my grants, um, is that I first of all, you apply to a lot more than you get. So there's plenty of grants I've applied for and haven't gotten. And um, it's just it can be really discouraging, but I think after the first failure, it gets easier. So you just have to keep going, right? Of like if you have one idea, maybe figure out the right place for it. And it's just really important to not be discouraged. Like sometimes you'll submit something, like I had something that I submitted to NIH several times. It was always reviewed terribly, and then ended up revising it and submitting it for a DoD Ovarian Cancer Academy Award, and that got funded. So it's just completely different on what people want to fund.
Right.
So it's really important not to take it personally. And I find that the best advice I can give about grants is that I got a ton of feedback from really experienced people. So people who can, who you can find people that get R01s at your institute, right? So people, I would look at people at GW who had, R01s from NCI for decades, and then ask them for feedback on my Ames page. One or two would then offer to read the whole application. If it's 12 pages, I don't want to make people read the whole application, but that's been really helpful for me because they have the perspective of experienced grant writers and reviewers.
Speaking of mentoring, did you have mentors to guide you with the transition during your first year as a junior faculty? And what was that experience like? And how long did it take you to get your lab up and running?
Yeah, anyone I asked for help was incredibly helpful. So it was a really positive experience. Everything just takes forever. It's like starting your own lab. I think the first experiment we tried to do, I had hired a technician. I came in January, hired a technician in February. She was great. We like tried to do an experiment and realized that we didn't have like the buffers. It's just the things that you think are lying around, like, oh, we can't do this experiment anyway. So it just takes a long time to get all the things ordered and in. But I would say we were doing experiments by March.
Oh, wow.
Not like, we do a lot of cancer cell culture. So it's basically you have the frozen cells, you thaw them, you can do experiments. So definitely no mouse experiments for like a year or so, but that was not planned. So that's okay. Yeah, so I think I really felt very supported. And it was more just it was really helpful to me for me to hire someone quickly who was high quality so that we could be a team, that she could really help me start all the ordering and get everything in place in the lab.
That's interesting. And as a junior faculty, have you had difficulties or challenges attracting students or postdocs to your lab? And how do you navigate that?
I think it depends for the person that you're recruiting, right? So I actually my plan was to start off with postdocs in my new lab because I figured that to get things up and running, having rotation students and early grad students, they have so many classes and things like that. I wanted to kind of begin with postdocs. So ironically, now I'm moving to more of a grad student lab, but I think that actually works well with the evolution of the lab. But so I listed 2 postdoc positions. I hired 2 postdocs in the first 8 months. So one joined in August, one joined in September. So I posted you post the job on the GW website. Many various applications came in.
Oh, that's great. Okay.
Yeah, some more competitive than others. So you can kind of narrow it down pretty quickly in terms of experience, publications, etc. And then I would bring people on campus to do a full day interview with my lab. So I'd have them give a talk on their PhD research, and then we would ask them questions, and I'd have them do one-on-one meetings. At that point, the lab was just me and Sarah, my lab manager. So we would each interview them, but I also asked more experienced PIs to interview them as well. And so I was like, do you have 20 minutes to talk to this person? Great. So to get their feedback. That was really helpful. So I was able to get 2 really good postdocs that way. One, to be honest, I had a connection with because he was coming from my graduate program at WashU. So I had reached out to them to see if there were recent graduates. So that was a lucky connection. So that was fine. And then the grad students are really, like the PhD students here are really great. A lot of them have experience before they've joined the PhD program. So everyone in my lab, I have 3 students now, each of them was either a lab manager technician for like a year or 2 after college or has done a master's degree. So they come in pretty experienced. And I found that that was really, really helpful for them kind of getting their projects kickstarted. They obviously do a rotation in their first year and then join the summer after their first year.
Got it. Now, as you're running your lab, are you able to make time to gain new research expertise in order to expand your research portfolio? Or do you typically rely on collaborations as needed?
Yeah, that's a really good question. I think that I can give 2 examples of areas that I don't really have a lot of experience in that we have kind of branched out, and both are collaborations, to be honest. So finding someone, so the DoD project, half of that project is working on antigen-specific T cells, and that's something that I have no experience generating or making these T cells. But luckily, both of my collaborators, Dan Powell at UPenn and Russell Cruz at GW, have a ton of experience in this. And so we've established a collaboration there, and my student who's working on that trains with both me and them, right? I've learned a lot, and that has worked very well. Another one that we're just starting is a collaboration on silencing HIV using CRISPR, which we're used to CRISPR and epigenetics, but not HIV. So that's with the collaborator, Dr. Bosca here. So he has all the HIV knowledge. So I think that collaborations are a good way to do that, although you have to, I think, be careful about not spreading yourself too thin.
And that's a great point. Speaking of collaborations, what's your approach to building new collaborations? Do you let it happen organically, or do you actively seek out opportunities to collaborate?
So I'm trying to think of the Ovarian Cancer Academy Award. I reached out to Dr. Powell because I knew him from going to ovarian cancer meetings. I thought he'd be a really great mentor and collaborator for that award. And what I think is really a good idea for any collaborative project, whether it's you're writing a grant to submit, you're generating preliminary data, or you already have a grant and you're working on the grant together, is to establish a monthly meeting. So like just take an hour every month on your schedule. It's on both of your calendar so that you can each present results, because otherwise sometimes things fall off the radar and everyone forgets. And so that was something that one of my other collaborators taught me. It's a really good idea. So that's how really we maintain them. Usually I just have the PIs do it. But for example, Dr. Powell gave a talk here in January, and I had my 2 students who were working on the project present for him and be critiqued by him on their work. So usually it's just PI's meeting, but then, twice a year or something, we have the trainees present too.
That's a great idea. I'll keep that in mind. Shifting gears slightly, can you expound on the cancer epigenetics research that your lab does?
Yeah, so we work on some pretty weird stuff. So actually, we work a lot on the non-coding part of the genome. So we know that our genome is very large, but only about 2 to 3% of it makes proteins, right? And the rest of it are things called repetitive elements, transposable elements, that are actually the remnants of some of them are ancient viruses that integrated into the genome a long time ago. So some actually as far back as mice or primates. Some are human specific. And then other elements that can jump around in the genome are about 20% of our genome. Most of these are no longer, they have not been conserved over evolution, and so they're no longer functional. So those are in our genome. They are normally epigenetically silenced. So think of it as putting a mark on the cells that would be a red light. When you have a green light means transcription, red light's going to turn them off. So we found during my postdoc that treating cancer cells with a drug that inhibited DNA methylation, and that's the red light that goes on these cells, that goes on these genes, will actually create an immune response against cancer cells. And we didn't know why. And so started looking into these old viruses. And it turns out that this drug activates the viral RNA. It doesn't make protein, doesn't make an actual virus, but the RNA that comes from these elements is recognized by the cancer cells as a virus. And the cancer cells then can decide to die, or they secrete chemokines that bring in the host immune cells to kill the cancer. So it really creates an immune response by activating these non-coding parts of the genome. Yeah, it's really interesting, and it's we don't know a lot about. There's so much we don't know about these repetitive elements and how they're regulated. They're quite different in cancer versus normal cells, which makes them really exciting targets, I think. But we need to learn a lot more about them.
Got it. And do you do the data processing and the analysis of the sequenced DNA methylation data in house, or do you rely on one of your uh, collaborators who has an expertise in bioinformatics analysis?
Yeah, I would say is absolutely not my expertise. I did some of it in my postdoc, enough to limp along. But I specifically hired my one of my postdocs, James, and he has some computational experience. I actually had him train because he was at WashU. I had him train with my colleague, Dr. Ting Wang, who really is an expert on repetitive elements in cancer, for a month or so to learn a lot of these techniques and analysis before he came to us because we had a collaboration already set up with Ting. So I wanted him to have some exposure to it, and then he's definitely learned a lot since he's come here. So um, he really represents the bulk of the bioinformatics knowledge in the lab. But then luckily, we now have all three of my PhD students can do RNA-seq analysis. And they've learned from him, and they've learned now in their classes they do more computational computational stuff than we do. So we like to do it in house because often if you are collaborating with someone, or even companies will analyze data for you now, right? I have no idea how to analyze repetitive elements. So we have very custom pipelines that we use. And so we really need to be able to do it ourselves. So I want to make sure, we have now, I think, 4 people in our lab that can do this. So I really want to keep that expertise going.
Well, that's impressive that you're able to do the bioinformatics analysis in house. Beyond that, how about the library preparation? Is that something you also do in house, or do you rely on somebody else?
Good question. Depends on the assay. For RNA-seq, we'll usually do it in-house. We do it all the time. We did do whole genome bisulfite sequencing for DNA methylation. For that, we hadn't done it before. And so we just sent the DNA to the company to do it and had a great experience with that. So I think if it's something that you've optimized and you're really good at, like do it in-house, like, for CHIP, for ATAC-seq, for RNA-seq, we do it all in-house to have control over that. But then if it's something you haven't done before, it's probably worth trusting the company to do it if you have a good reference on the company from a colleague.
Got it. Now, looking back at your academic career up until this point, are there things that maybe you wish you had done differently? Or perhaps you're glad that it worked out the way it did?
Yeah, I can't think of anything I would have done that differently. I'm trying to think. I definitely had points where I wasn't sure whether or not I should attend a meeting or, for example, bring my students or trainees to a meeting. I always decided to do it, but I am very glad I did, especially now that we haven't been at meetings for a year, right? So I so appreciate it. So if you ever are worried about the cost, that's what I would say. If you're ever worried about the cost of attending a meeting for you or paying for your trainees to go, just do it, especially at the beginning. Because I think that the benefit is so much greater than the cost. So that's my one piece of advice, because I've gotten so much out of attending these meetings. You just don't know who you're going to interact with. I literally have collaborations because of these meetings. And I think for the trainees, they've gotten really important feedback. So don't let the cost, which is significant, like I completely realize how much it is, but it is worth spending the money. When you think about, how much you spend on your science every day, it is worth spending the money to go to the meetings. So I that's not like a regret, but I definitely like thought about it a couple of times of like, wow, should I be spending thousands of dollars on this? Like, I'm not sure it's worthwhile. I'm trying to budget. But I'm really glad I did. So whenever we get back to going to meetings in person.
Yeah, and I'm hoping that happens sooner rather than later. Now, going forward into 2021 and beyond, what are some of the things that you're most excited about?
Yeah, I think that there's really a lot in this space that we still need to learn. It's an exciting area to be in. And Yeah, I think that I'm not that concerned about it. I think that there's a lot we don't know. So hopefully I get, the R1 that scored well. That would start in April. And looking forward to kind of starting to we have a paper interview at Cancer Research. So getting our, now we're okay funded, but now it's more of making sure we get these manuscripts through review and published are the most important things.
Right.
And I do my mid-tenure review with my department this spring. So
Oh,
what we do at GW is you have, like, in the midpoint of your tenure process, you put together a fake tenure dossier that has, your CV, your teaching service, etc. And your advisory committee gives you feedback on, what's going well and what should be improved by the time you apply for tenure. So at the end of that time. So I have that coming up. So that'll be really important in terms of getting good feedback.
got it. Well, Kate, thank you so much for taking the time to come on to the Catalysts podcast to share your perspective with our listeners. I really appreciate it.
Great. Thank you for having me.
With that, we will wrap up this episode of the podcast. If you liked what you have heard, I encourage you to share this episode with a friend. I'd also love to hear from you and how you are striving to be a catalyst. So I invite you to leave a comment on my Twitter or website. This is your host, Kedar Aris, signing off. Thank you for listening, and until next time, connect, discover, transform.